Namaskar. I am Alka Arora, working as a principal scientist and professor computer application in the division of computer application at ICR ISRI. Today we are going to talk about AI application in the area of plant phenotyping. As far as the plant phenomics is concerned, this is the study of plant growth and performance based on morphology, physiology, and the phenotypic traits or the characteristics of the plant. Basically, there are certain parameters. For example, we can talk of leaf area estimation, we can talk of senescence, number of spikes, leaf area. So all these phenomics parameters, they play a very important role as far as the plant growth is concerned. When we talk of conventional measures to basically measure these traits, so either these conventional methods are labor intensive or they are prone to error when we are talking of dealing with the large amount of data set. Plant phenotypic. So this is an area which is emerging, which deals with application of AI algorithms on the data, which is related to crop health, growth pattern. And then these algorithms basically they analyze the images. We are talking of the image data which are captured with the help of various sensors. So there are parameters which are identified with the help of this image analysis. So these parameters are your stress, your disease, identification of nutrient deficiency, and at the same time there are other factors also which play a very important role as far as the crop performance is concerned. So all these parameters, estimation of these parameters with the help of image analysis comes under the arena of plant phenotyping. So this is a glimpses of the applications of deep learning when we talk of plant phenomics in the area of agriculture. So you can see here when we talk of the conventional methods, so when we talk of the leaf area estimation or identification of the different parameters, so you can see all these are very labor intensive and at the manual process. But when we talk of coming to similarly to find out the relation between the genotype and the different phenotypes, so this is very time consuming as far as the traditional methods are concerned. But then in the area of plant phenotyping, nowadays the concept of high throughput phenomics, which is gaining importance, you can see here there are different platforms, or we can say there are different sensors with the help of which the data is collected. So these sensors, we can have the RGB cameras, we are having the spectral resolution cameras, we are having thermal cameras, LiDAR sensors, and then there are different platforms using which the data is being captured. So all this data is being analyzed with the help of a big data platforms. We can treat this large amount of data because nowadays the computation cost has gone down and the processing power of the computer has increased with the emergence of the new technologies. You can see, so this is the various phenotypic data which is being collected with the help of these sensors: leaf area index, chlorophyll identification, biomass estimation, nitrogen deficiency estimation, the height, width, and the yield estimation, which is related to all these phenotypic traits. So all this can be calculated, all this can be computed, and then with the help of combining together this genotype and the phenotype uh, fundamentals, we can do the genomic studies, and then combining these two together, one can go for identification of the varieties which are tolerant to, you can say, different type of stresses, drought, heat stress, and we can identify varieties which are sustainable in nature. So giving you scenarios in which our division has done some of the studies, some of the case studies in the area of plant phenomics. So we have done work in certain projects as well as with the help of our PhD students. So all this work has been carried out in our Nanaji Deshmukh Plant Phenomics Center, which has been established at IRI. There is a company called Lemnatech who has established this infrastructure in this particular Nanaji Deshmukh Plant Phenomics Center. So these are some of the screenshots of the Phenomics Center. In this, we are having different type of sensors available. There are RGB sensor, NIR sensor, infrared sensors, very near infrared sensors, and the fluorescent sensors available. And these studies has been carried out with the help of this high throughput plant phenomic facility. Image analysis has been done applying certain deep learning algorithms to extract several phenotypic parameters which are related to plant growth. So these studies has been carried out using RGB images, which is of the nature 2D, and at the same time, sub studies are being carried out, which is being done on the 3D imagings also. So we are going to see these studies. Now coming to the very first study, which we are going to talk of today, that deals with detection and counting of spikes in a single plant. This case study is related to the wheat. In this particular case study, this is divided into basically two steps. The very first steps deal with the identification of spikes. So in this case, our region of interest is spike, which we are considering as an object. On this identification of object, basically spikes, there is a deep learning algorithms, which is the convolution encoder decoder deep learning method, which has been applied, and based on that, there is a model which has been created, which we have given the name spike segment. So this spike segment model basically deals with the identification of spikes from the wheat plant, and then in the second stage we are taking care of the counting of this particular spike in order to calculate the yield. So this is how this spike segment model is working. In this model, basically we have considered the RGB images of the size 1656 into 1356. You can see this is a very big size as far as the image processing is concerned. So this particular image, for this particular image, our aim is to identify the spikes. Therefore, different patches are being generated from this particular image. Why these patches has been taken into consideration is because our aim is to extract the features which are related to the spikes. That's why in order to have the learning, efficient learning, we'll say, this identification or this patch generation work has been considered. So this image has been divided into different patches of the size 256 by 256 in order to learn more number of features from the images. Then we have given it this network, we have given a name LPNet, which deals with the local patch extraction network. So this is local patch extraction network basically deals with identifying the patterns, or we can say the spike here. Then on this particular LPNet, we have applied this encoder decoder model, and then we have gone for combining together the merging of the mask patch images. So you can see here, so this is the binary image. So this is our region of interest object. This is our spike. So these spikes has been identified into different patches. Then we have gone for merging of this each mask which has been generated, and then again resizing the same into the 256 by 256. Then another model has been devised on top of it, which is our global mask refinement network. In this, these masks are being refined and then converted back into a single image. So you can see this way the network is learning through a series of events, and when we have applied this model for the computation, or you can say the identification of spikes, we got more than 90, almost 95% accuracy as far as our spike segment model is concerned for identification of the spikes. So in the second stage, when we are dealing with the counting of number of spikes, so in this particular case, we have taken the images of the same plant from three different directions. For example, 0 degree from the front, 120 degree and 240 degrees. On all the three images, this has been passed through our spike segment model, and at the end what we are getting is this binary mask, which is related to our spikes. Then another algorithm, which is the flood field technique algorithm, which is used for the object counting. So the same has been applied and the spike has been counted. So the very purpose of taking images from three different directions was we can consider, so if there is certain overlapping happening, so that can be considered. You can see the maximum number of spikes which has come out is 12. So with this, we have considered 12 is the maximum number of spikes which are available in this particular plant. Combining together this complete approach, for which for the spike secret model we got the accuracy of almost 95%, and then as far as the counting of spike is concerned, we have applied certain algorithm flood field technique and we got the accuracy of 92%. Now we are going to talk about another study which is on the rise, which deals with the detection of pedipenical stages. So in this case, we have gone a step further in which we are dealing with the different stages identification. So you can see, because all these stages are interrelated, so this has been created into, let's say we have considered 5 significant stages of the paddy growth based on the panicle. So these stages are your booting stage, heading stage, anthesis, grain filling and the grain maturity stage. So again, the data for this particular study is also collected from the Nanaji Deshmukh Plant Phenomics Center. We have considered the RGB images and then we have considered the images from the 4 different directions. Because the image size you can see here is very big. So you can see 4384 into 6576, because for certain stages, for certain objects to be identified, we need to take images with the uh, which is very big in size. But then we have faced certain challenges also, because the images were too big to process and there were too much background which need to be taken care of. So we have done the cropping of these images and resizing it to a certain level so that our object identification can be done effectively. So we have resized it into this particular scale, and then this analysis was carried out. So this is the pre-processing stage that has been done. So on this, different deep learning models were applied to see the accuracy for the identification of the different panicle stages. For some of the stages, we got the accuracy more than 90%, but for two stages, we got the accuracy almost in the range of, you can say, 70 to 75% also. So you can say, so this is the accuracy we got when we applied different deep learning models. So this is the faster RCNN, SSD. There are different versions of YOLO which was applied, YOLO V5, YOLO V8, and then different versions are available now. Our approach, our model, which we have given the name Panicle Date, which is an ensemble model based on the ensemble of YOLO 5 and YOLO 8. And then combining together, we got the accuracy of 93%. But at the same time, you can see here in this particular range, 0.5 to 0.95 range, we got more accuracy as far as our other models are concerned. That's why we can say this panicle date is giving a very good accuracy as far as the different stage identification of the panicle are concerned. And then at the same time, after the panicle identification, attempt was done for the yield estimation also. So with the ground truth data available, the regression analysis was carried out, and then we got a very good accuracy as far as the yield estimation is concerned. I'm going to share another case study which is related to the counting of seeds, or you can say counting of mustard silics. So we know that the silics, they play a very important role as far as the mustard genetic improvement program is concerned, and at the same time the yield aspect is concerned. So for this particular aspect also, the data is generated from the Nanaji Deshmukh Plant Phenomics Center IRI, and then on top of this data, certain pre-processing techniques were carried out. And then the YOLO model has been used for the automated detection of the silics. on the mustard plant. So you can see this particular software which we have given the name music, mustard select. So in this particular software, you can open the image file of the mustard plant, and then it will give you counting of the selects, and it will show you the presence of the selects on this particular mustard plant. We got some accuracy, more than 60% accuracy for this particular thing, and we are working on improvement of the same. Another case study which I am going to discuss here is related to senescence estimation or the senescence quantification. So we all know that senescence is when the plant changes color during the different stages. So automatically also or naturally also the plant changes color over a period of time during the different growth stages. But at the same time, when the plant is exposed to certain stress condition, in that conditions also there is senescence which is appearing, or this is unnatural or untimely basically yellowing of leaf that happens. So that is called induction or occurrence of the senescence. Again, this method, when we look at it at the conventional method, so it is very prone to error, but at the same time, when with the help of image analysis, this is we can estimate it in a very good manner. So for this particular study, we have considered different machine learning algorithms. So these are the different machine learning algorithms which has been considered, like naive Bayes, K nearest neighbor, decision tree, random forest, the gradient boost, and the artificial neural network. So the data collection was done, and at the same time pre-processing was also carried out. So how the data collection was done? For each class, for example, for the senescence purpose, there are 5 classes which has been chosen for the senescence and for the greenness part. So these classes are brown, yellow, pale yellow, dark green, and the light green. So these 3 classes, like brown, yellow, and the pale yellow, they are contributing to the senescence, and dark green and light green are basically our other categories. For each basically classes, the data or the pixel values, the image pixel values were considered, because your RGB images are made of the pixel, and every pixel has got certain intensity which basically reflect or which gives you the color of that particular pixel. So this is a combination of your RGB values. So we have collected almost 1000 pixels for each color category, and then we have considered certain background categories also. For this background categories also the pixel samples were taken. Then how this senescence estimation was taken into consideration. So this is the flow chart, because the complete input image has been considered, which was passed to our models, where the pixel by pixel reading of the image was done. Every pixel was considered, which has passed through the different machine learning algorithms, which we have talked in the previous slide. And then it has gone for the identification of different colors. For example, if the pixel belongs to a green category, so the green pixel variable will be incremented by 1. Similarly, all the pixels were divided into different classes, and then with this we have gone for the estimation of the total plant pixels. So which is a combination of your, you can say, the complete plant area which has been calculated, which is a combination of all these pixels which has been divided into different color combinations, and then total plant area was calculated. Then when we have applied different machine learning models, so we can see here many were giving good accuracy, but at the same time ANN was performing best with the almost 96% and 97% accuracy for the classification of pixels into different categories or the different classes we have considered. So you can see here, for each class the binary mask was generated, for example, and then we have given, because all there are 5 classes, and then for all class, the binary mass was generated, and we want to represent it or want to show it physically. That's why this binary mass was given certain colors for the better identification. So we have given yellow color for the pale yellow pixels, blue color for the light green pixels, green color for the dark green pixels, yellow color for the yellow pixels, and red color for the brown pixels, so that this senescence can be easily identified. So then all this, you can say, the binary images which were given a particular color, they were combined together to make a complete picture. And then you can see, with the help of this software, we can see this colored mask of the plant from initial stage to the maturity stage. You can see there are 18 stages which was considered. You can see here how the senescence is appearing into the plant at different stages. So initially it is all green, and then at certain stages the senescence has started appearing, so which we can easily relate to when this particular plant was introduced to certain kind of a stress, drought stress or the heat stress, and then at what particular time the senescence has started coming into the normal plant and into a stressed plant. So you can easily view the different plant stages, and at the same time the complete plant area and areas into different senescence classes was also calculated with the help of this particular software, and we can download all this information into an Excel file also. Another study we are working with that deals with the identification of Cepheid grains using your hyperspectral images. So this moisture basically plays a very important role as far as the yield is concerned. So this is an advanced study which we are working, and this hyperspectral images that enables for the detection of your Cepheid grains. In this particular scenario, the wavelength, which is around 1415 nanometers, so this mainly contributes towards Cepheid grain detection. So you can see this is the certain years, certain kernels which were removed, which is shown in the gray, which is empty, and this is the black one, which is filled. So a model prediction from the hyperspectral images which was done, and you can see we are able to identify certain this thing. Still we are working on this particular approach, and another approach which we are working now, which is we have considered or we have seen there are certain challenges we faced as far as the 2D images are concerned. So we have started working with the 3D imaging concepts also, with the assumption, because our 3D images, they accurately present the spatial information between the objects, allowing viewers to perceive depth as well as other parameters. For example, when we were considering the 2D images, we have considered the images from the 3 or 4 different angles so that we can cover all the sides. And at the same time, when we are considering the 3D imaging part, so we are expecting that this will give us the more depth information. So this depth perception basically gives, or you can say it adds up to the visualization aspects, and at the same time we are hopeful to get the better results as far as the phenotypic parameter estimation is concerned, because mainly it is related to your visual aspects. And then at the same time, because nowadays a lot of cameras are also available, for example the LiDAR camera which are available in the market, with the help of which you can, and there are other cameras also, with the help of which you can basically click your 3D images, or at the same time there are certain technologies, certain methods which are available using which you can convert your 2D image into the 3D image also. So we are working on both these aspects. But at the same time, now to conclude, I can say there are different areas we look forward to work or maybe for the future collaboration aspects also. So computer vision in plant phenomics involving high throughput of the different types of images, because mainly we are working with the RGB images and we have started with the hyperspectral images also, but there are different sensors available in the market nowadays, and we want to try out using all different type of sensors, computation of the different phenomics parameters. At the same time, this 2D and 3D image modeling, extending the results from the phenomics lab to the field condition, prediction of yield for all the major crops in the lab condition, and at the same time evaluate more phenotypic parameters, for example biomass, leaf angle, because they can also again add up into different um, parameters estimation, or at the same time maybe they can contribute towards the yield estimation or overall plant growth aspects, and the application of the 3D technology in the phenotypic parameter estimation, AI application to detect nutrient deficiency in major crops, prediction of yield using drones in the field conditions. So we want to take up our uh, models to the drone level also, detection of Sheffy grain in the cereal crops in which we are working, analysis of the seed quality, counting of the seeds, identification of different stress tolerant genotypes. So these are some of the areas in which we are working, and we would like to take our collaboration further. This work has been carried out with AI infrastructure which is available at ISRI in the ICR data center. We have one NVIDIA server, NVIDIA DGX server. So all this processing has been carried out using this particular system. These are some of the references of the work done which we have presented in this particular presentation. So mainly related to, you can look for certain some of these references and get more benefit out of it if this area is of interest to you. So with this, thank you so much